Hello everyone. Today we'll discuss the part 56 of the important notes for biochemistry. Alcohol has zero order kinetics. There is no negative feedback control for alcohol metabolism. Ethanol and fomepizole, they are used in the methanol poisoning. Alcohol or ethanol actually causes fatty liver due to increased NADH by NAD ratio. I have made a separate video on the fatty liver, so let's check that out. Protein precipitation can be done by heat, strong mineral acids, heavy metal salts, and also alkaloidal reagents like trichloroacetic acid, sulfosalic acid, phosphotungstic acid. Also, organic solvents like ether and neutral salts like ammonium sulfate can cause protein precipitation. Radioimmunoassay is used to assay the drugs, vitamins, hormones, and also to analyze the anti-DNA antibodies in the systemic lupus erythematosus. Most commonly used method for the HbA1c is the ion exchange high performance liquid chromatography, that means ion exchange HPLC. The best method for HbA1c is the enzymatic assay, that means over here horseradish peroxidase enzyme is used. Negative charge in the fibrinopeptide A and B is due to the acidic amino acid, that is the aspartic acid and the glutamic acid. Bacteria in the yogurt actually produces a form of lactase enzyme which breaks the lactose. So that's why curd doesn't contain lactose and so can be given to the lactose intolerant patients. Type 4 collagen, which is present in the basement membrane of the glomerulus, is defective in the Alport syndrome. Type 7 collagen, which is present at the junction of the dermis and epidermis, is defective in the Epidermolysis Bullosa. Fish odor syndrome or the trimethylaminiuria, it is actually a rare metabolic disorder due to the absence of enzyme trimethylamine oxidase, that is the flavin monooxygenase, and this particular enzyme requires vitamin B2. Sources of trimethylamine are the dietary choline, trimethylamine oxide by the bacteria, egg yolk, liver, and fish. Trimethylamine is normally oxidized in the liver by the enzyme trimethylamine oxidase into trimethylamine oxide, which is odorless, and this is excreted in the urine. But if this enzyme is not there, then the trimethylamine is excreted in the urine, which gives rotten fish-like odor to the saliva, sweat, and urine. Chaperones, which are located in the rough endoplasmic reticulum, are used in the protein folding. For example, immunoglobulin heavy chain binding protein, glucose regulated protein 94, GRP 170, GRP 78, calnexin, calreticulin, protein disulfide isomerase, peptidyl prolyl cis trans isomerase, HSP 47 or the heat shock protein 47, and the endoplasmic reticulum protein 29 and the ERP 57. Calbudin is a group of several calcium binding proteins. Most abundant pro form of vitamin A, it is the beta carotene, which gets broken down to 2 molecules of retinal. The secretory vesicle are the vesicles that mediate the vesicular transport of substances. For example, hormones or neurotransmitters from an organelle to specific sites by fusing with the cell membrane to release its contents. Subacute combined degeneration of the spinal cord, that is known as Lichtham's disease. In this, there is degeneration of the posterior and the lateral column, and it occurs most commonly due to the vitamin B12 deficiency, but also it can occur in the vitamin E or copper deficiency. Product of the phosphofructokinase 1 enzyme actually activates the pyruvate kinase, and this is known as feed-forward activation of the glycolysis. The only kinase in the body which is reversible, it is the phosphoglycerate kinase in the glycolysis pathway. Enhanced uptake of the glucose by the cancerous cells due to the Warburg effect can be easily seen by the PET scan or the positron emission tomography. There is presence of a mitochondrial bound hexokinase in the cancerous cells which is resistant to the feedback inhibition. So today up till here, I hope you found this helpful. Thank you so much.